Hey everyone, welcome to Think Science, and if you haven't been on our channel before, like and subscribe for more biology content. This is the third video in our biological macromolecules segment, and we're jumping into nucleic acids after carbs and lipids. Be sure to check out those videos as well. Before we get started, let's begin with the question of the day. DNA is a nucleic acid, and it's used in forensics to investigate crime. What does the acronym DNA stand for? Leave your answer in the comments below. So, nucleic acids encompass DNA and RNA, and primary functions include information storage and gene expression. In later videos, we will discuss how exactly DNA stores the cell's genome, and how different types of RNA express it through a series of biological processes. However, for this video, nucleic acids are molecules made out of carbon, hydrogen, oxygen, nitrogen, and phosphorus. There are two main parts to nucleic acids, the sugar-phosphate backbone and the nitrogenous bases. We'll start with the sugar-phosphate backbone. Here is a picture, and as the name suggests, to each sugar molecule, there's a phosphate group attached. The sugar can vary based on the type of nucleic acid. RNA, for example, has ribose sugars in its backbone, whereas DNA has deoxyribose sugars. Notice the prefix deoxy. All this means is that a deoxyribose sugar does not have an oxygen atom attached to the second carbon of the sugar ring. The second part of the nucleic acids includes the nitrogenous bases. These are a total of 5, adenine, guanine, and cytosine, which are all found both in DNA and RNA. The last two include thymine, which is specific to DNA only, and uracil, which is only found in RNA. One nitrogenous base is attached to one sugar from the sugar-phosphate backbone we discussed earlier. One nitrogenous base bonded to the sugar and phosphate group makes up one nucleotide. Because all of these nucleotides are attached by the sugar-phosphate backbone, the entire chain of nucleotides makes up a nucleic acid, like DNA. Nitrogenous bases fall into two categories. There's purines and pyrimidines. If the nitrogenous base has two rings, it is a purine. If it has one ring, it's a pyrimidine. The purines are adenine and guanine, and the pyrimidines are cytosine, thymine, and uracil. Here is a typical DNA helix animation you might see in one of your biology classes. In humans, DNA is double-stranded, whereas RNA is single-stranded. This means that DNA has two sugar-phosphate backbones, and this is because nucleotides attach to one another via nitrogenous bases, forming a double helix. The nucleotides in RNA also attach in the same way when carrying out certain biological processes, but that will be discussed in later videos. If you look carefully, the same colors always pair together. Adenine always pairs with thymine, or uracil if we're talking about RNA, and cytosine always pairs with guanine. Also note that the DNA is always uniform in width throughout its entire length. This is because a purine is always paired with a pyrimidine. Therefore, the width will always be 3 rings long. A purine with a purine would be too short for the DNA gap, and a pyrimidine with another pyrimidine wouldn't fit in the gap. Make sure to like this video and subscribe to our channel Think Science for more science videos. If you have any questions about nucleic acids, be sure to comment and we will do our best to answer. Thank you for watching Think Science.